This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 739, recorded on April 2nd, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. It's, what's the temperature now? It's freezing. It's 32 Fahrenheit, 0 Celsius, and we're having actually some scattered flurries.
Wow. Funny weather. Funny weather.
It's really kind of cold and blustery.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing? And I don't even know what the weather is here. It's, hang on, I'm looking it up. I've almost got it, 69 degrees, partly sunny. So perfectly fine day.
And from Madison, New Jersey, Brienne Barker.
Hi, it's great to be here. I'm happy to be inside because it's sort of similar to what Alan was saying. It's 34 and very cloudy here.
Our guest today is from Baltimore, Maryland. He's at the Johns Hopkins University. Arturo Casadevall, welcome to TWiV.
Thank you, Vincent. It's cold here too, but not as bad as Alan and Brienne are saying.
Well, where you are in outer space, it's really cold, right?
Oh, yes.
Unless you're in the space station.
It's about 4 Kelvin out here.
We actually interviewed an astronaut on the International Space Station last year. And she said it's always 70 and clear here, which was very funny. So this is your first TWiV. You've been on TWiM before, Arturo.
Right. I think we've done once or twice. We did one at the general meeting a few years ago. It was a lot of fun. We talked about the problems of science.
Oh, yes. The problems of science and science publishing. And then we were on another one before that with
on fungi or something related to fungi. We did another ASM meeting.
So Arturo, you spent many years here in New York at Albert Einstein, right? You were chair of micro, is that right?
Right.
And you did work on fungi, right?
Well, actually depends who you ask. I mean, I'm one of those people that people have trouble with. In science, one of the real problems in science is that scientists like to pigeonhole somebody. They don't like to call somebody else a scientist. Well, that person is a virologist. No, it's a real virologist. No, it's an immunologist who plays with viruses. And instead, I think that does a lot of disservice. But you're right. I think that I trained with Matty Shar for my postdoc, and I don't primarily. One thread through my entire career has been antibody-mediated immunity. Applied to the fungi, but a lot of basic immunology. But if you ask an immunologist, they think I'm a microbiologist. The microbiologists don't know what to do with me. And, we just manage.
We often talk about the fungi growing in Chernobyl. You had published a paper on that, remember?
That's right. Well, actually, I think that was one of the probably in the big picture, one of the most important things we've done. What it showed was that melanized fungi can, by some manner, capture radiation and for growing in food. And by the way, you're talking about the space station. The space station has been overrun by fungi, which is in the high radiation environment of space. The fungi tend to grow faster.
Wow. And those came with the people who brought it there, right?
Actually, it comes with every pot that once you open up the air, all the fungal spores go right into the space station.
Is that a problem for them?
It's a problem because if you go to the internet and you Google, fungi in space or something, you see mold growing on the electronics, mold etching the glass of the space station, melanotic fungi growing on the walls. But to tell you, NASA is very interested in this because melanin is a strong, it can shield against radiation. So one of the great problems of any interplanetary travel is going to be radiation. And this way, all you really need to do is send a spore up to the space station, and you can grow your mold on the outside. So you can imagine future spaceships having mold, black mold, all over the outside, providing radiation, and you could feed the mold with the garbage from the astronauts.
So future spaceships will be made of fungi.
I am not kidding, Alan. We are having those discussions, and they are in fact funding some of this work. They are very interested because it's a lot easier sending up a spore that can grow from human garbage in a space station than sending up lead.
Sure.
No.
That is so cool.
So how's Baltimore treating you, Arturo? Are you happy there?
Yeah, I'm very happy here. I think that from a point of view of career transitions, it was very good. I came here because I wanted to do something about the problems of science and launch a new PhD program. And we have accomplished that, and it's very popular. And we try to teach scientists how to do good science from first principles, critical thinking. You know, in my entire career, no one ever said to me, Arturo, if you ever do an experiment and you get a result, you need to do a dose response. No one ever said that to me. Well, what was I supposed to pick that up? From the bench? By osmosis? So now, right. No, you know, and I had very rigorous training also in physical biochemistry. Nobody ever said that to me. Nobody, this is the kind of thing that you want to teach from day one to a scientist. How do you approach data? How do you approach error?
Well, Arturo, part of the problem is a lot of the training is not codified anywhere, right?
That's right.
You depend on your mentor to tell you, and they don't always tell you the right things.
Every PhD experience is different. Every lab is different. But we think that after 250 years of the scientific revolution, we should be able to teach people some first principles. You make a measurement. What do you do with it? You know, certainly a dose response. What is a control? You know, what are the logical errors? You know, how much of our science is built on logical problems? So, for example, in the field of immunology, I can tell you that when I got into it, that antibodies didn't protect against fungi. Why? Because the experiments have been negative. So they use negative data as a positive inference. If you do an experiment and it's negative, all you know is that it's negative. You know, but we have entire fields and entire dogmas built on, you know, that's not the way it is because the experiment was negative. Stop it.
Well, Arturo, earlier, sometime last year, we never thought coronaviruses did antigenic variation. And you know why? Because no one ever looked.
Well, because there was nothing interesting about coronaviruses, right?
Right.
Yet we make dogma based on nothing, right? So
right. But, you know, it was an RNA virus, and it should have been, it needed to be there from the beginning, from first principles. If you have RNA, are you going to replicate that? Error prone?
That's why I like having you on, Arturo. You're always interesting and provocative, but you also have these big picture views of science, which I think all of us need to have. We tend to get focused, right? And that's not always good.
So I advise everyone to learn something outside their field every day and spend no more than 5 minutes in it. Read something outside the field. And what happens is it's a much better life. The world is much more interesting. And you can make connections. You know, in our lab meetings, for example, we discussed the physics of pizza. There is a book called The Physics of Pizza. Why do you take a pizza from the crust? And why don't you bite into the other thing? Because you burn the roof of your mouth. It turns out the crust is at the same temperature, but it doesn't burn you. So then you can talk about heat transfer. You can talk about a lot of things. And the experience becomes even better than just eating your pizza.
There was a series of published letters a number of years ago in Nature on that subject that I think may have founded the field of pizza physics.
Of pizza physics. There's a book on it. And we do it. And we try to enjoy every aspect of this. You can learn a lot from a slice of pizza.
Well, here on TWiV, we try to think outside of the viruses that we are used to. You know, at least we try that. So it's something. You know, if we talk about physics, then our listeners say you don't know what you're talking about. So
I don't know what I'm talking about. So you can just save that criticism. I'm happy to plead guilty.
Yeah. All right. So today we have you to talk about convalescent serum and COVID-19. And I just heard you say a while ago that you've always been interested in antibodies. So I guess that's where it comes from, because I was going to say, you know, I know early in this outbreak, I heard your voice saying we need to do convalescent serum. So I was going to say, where did that come from? And I guess it's from your background, right?
And my interest in history. I spent a lot of my time reading the past. And when I first went out as an investigator, I got into a typical fight with the study section. And basically what they said to me was that the animal model needed to reproduce disease for you to make the inference that antibody immunity worked. And the way that I dealt with that was go back into history and show that serum therapy had been developed with animal models that had nothing to do with disease. Mm. For example, pneumococcal pneumonia is a respiratory infection. They worked out the serum that they used in a mouse intraperitoneal model. Nothing to do with it. In fact, that's how they standardized the serum. So what I did was I wrote a paper, published it, and then I threw the weight of history at the study section. And eventually I got funded, but it didn't help right away because all I did was make them more upset.
Yeah.
But I didn't quite learn that. I was very young at the time. Yeah. I should have rephrased the issues differently, but I knew this enormous history. And I knew that up to the 1940s, antibody had been used for many things. And when the epidemic first started, people were talking about monoclonals, antivirals, vaccines, but nobody was talking about plasma. Mm-hmm. And what I did was I wanted to get the word out, and I didn't know how to do it. So I decided to write op-eds. So I finally got an op-ed in the Wall Street Journal, and that was key because they rewrote everything in 650 words. They got the message out, and then we were able to organize very quickly on the basis of an op-ed.
Mm. So I my first encounter with convalescent plasma therapy, and I don't know if you know this story. Did you know Jordi Casals?
I know the name, yeah.
So he was a virologist working at Yale, and when the first Lassa patients came from Nigeria, one of them was brought here to Columbia actually, and they saved her serum. She recovered, and then he got sick working on the virus, mouth pipetting, and they gave him her serum, and he survived.
Yeah, he was treated early. That's
the key.
So is key. So as of today, 500,000 people have been treated in the United States with plasma. The data are mixed. Why are the data mixed? Because in many of the clinical randomized controlled trials and much of the work, they've forgotten history. There are 3 principles to antibody therapy. And in fact, they were never written down because they were so well known that they were obvious. I just had to write a paper in Bio that just came out listing that. So what these principles are, the preparation must have antibody to the agent. 2, that's the first principle. Some of the clinical trials have used plasma that in retrospect had no antibody because they couldn't measure it at the time. And by the way, the Ebola trials in West Africa had the same problem. Because what happens is you can't look at antibody often in the middle of the epidemic. You don't have the assay. So they say, hey, Vincent, did you have Ebola? And you say, yeah, I recovered. So I said, give me your arm. And it turns out that you had something else. So principle number 2 is that there has to be sufficient antibody. And principle number 3, which is the most difficult for physician, is what I call the temporal principle, that it must be given early. And look, anybody who knows anything about the pathogenesis of this disease will agree. It has a viral phase. Plasma works as an antiviral. In some people, this viral phase triggers an overexuberant immune response that puts you in the intensive care unit. Antibody is not going to reverse inflammation.
Yeah.
So that's the crux of the issue.
So I have a question. Oh, sorry. Go ahead.
Yeah, you're good. Go ahead.
So related to what you were just talking about, I've kind of wondered, is there any principle about how far into convalescence the donor needs to be? I've wondered about things like whether there might be inflammatory mediators that were in the convalescent plasma, unless you get someone who is, later in their recovery phase. Are there any principles about that part?
So Brianna, you're absolutely right. and your insight is correct. Convalescent plasma from people who recover with COVID, it has often the inflammatory mediators. And what is scary, because these people have recovered, and we're not even talking about long COVID. That paper came out of Hopkins here. We published it a few weeks ago, which means that even when people are well, they have some indications of the inflammatory fire still going on, the smoldering thing. But we don't think that that's the active agent. And the reason for that, Brianna, is because a study was done that was just published in the New England Journal that I was involved with, showing a dose response with antibody. So if you give more antibody, the person is more likely to survive than if you give less antibody.
And you
so
mean
if you take units and you titer them, right?
Right.
From everybody who recovered, and you would think this involved thousands of patients, and you would think that if it was the IL-1 that was in the background or something like that, that we were not measuring for that. We were measuring for a dose response to antibody. And that's a very powerful piece of data.
Yeah. So, yeah, I think you sent us that to look at, and I hadn't thought about that aspect of it, because you're giving a controlled amount of convalescent serum and different amount of antibody, but you would expect the controlled amount of serum would have the same.
On average.
That makes perfect sense.
Yeah.
For the naive listeners out there, could you briefly go through how you make convalescent plasma and what sort of assessment that you do of it to determine, whether or not it might be good or bad, and what sort of risks might be involved?
So let's do the first thing. So convalescent plasma, by definition, comes from people who recover from disease. So you need to be recovered, and you ideally need to be at least a month out. That's because some people carry their PCR positive for a while, and you don't want to bring anybody into a transfusion facility. And then they donate. And then that plasma is tested for all the known pathogens that could be transferred by the blood, the hepatitis, the HIV.
So plasma means you take blood from a person and you centrifuge out all the cells?
Richard, that's a great question. That's serum. That's the way that it used to be made. It used to be in the old days, they will take your blood out, they spin it down, and they take the serum. The difference between serum and plasma is that plasma has the anticoagulation agent factors. So the way they do it right now is they hook you onto a plasmapheresis machine. You sit there for a couple of hours, and this removes the liquid, the antibodies, plasma of the blood without removing the cells. So people don't lose any red cells or anything like that. So then that fluid is collected. By the way, a single person can donate 2 to 3 units. So one person can help 2 or 3. Plasma therapy is the only therapy that is made in people. And then you take that thing and you bring it into the lab and you check it for antibody content. And there are a zillion assays. You can also check it for neutralizing antibody content. There are pseudovirus assays, and it's being validated, by the way, in BSL-3 type of virus. So then you take your units and you label them. And then you can freeze them for up to a year. Mm-hmm. And then the doctors write an order, and then this gets distributed within hours, and then patients get it.
Am I correct in assuming that because there are no cells, there are no issues with blood type matching or anything like that?
You are largely correct, but the blood banking industry is extremely careful. So they don't even take a risk on that. They match the donors just in case there are some antigens there to try to reduce any sort of transfusion reaction.
Okay.
So is it the blood banks that are involved in collection of this?
Yes. This is a blood banking procedure. In New York City, for example, the New York Blood Center has done a lot of the collections.
Well, and so this plasmapheresis process you're talking about is in fact a routine thing that the blood banks were already doing for other purposes.
And the United States had the capacity to produce millions of units before, because plasma is used in surgery, often when there is a bleeding problem, to provide bleeding factors. So plasma was already established therapy without being convalescent. All that you do here is that you adapt it to take it from those. But I can tell you that a year ago today, the big concern was antibody-dependent enhancement. Mm-hmm. And when we first started this, I can have emails from some of the most eminent virologists, an immunologist that says Arturo, and they're sending me the links to other coronavirus papers where previous experiments have shown that in some cases coronavirus can induce ADE, as you know. And so we were extremely concerned in the first few infusions. And when the first patient was treated on March 27th of last year, I didn't sleep all night. I kept looking at my phone, thinking that I was going to get an email that the infusion of plasma resulted in a cytokine storm, because you can imagine that this could have gone the other way, right? And it turns out that there has been no signature of ADE whatsoever. Mm-hmm. And that's incredibly important, because had there been any issue with plasma, it would have slowed down the vaccines.
Yes.
Yeah.
And it would have slowed down the monoclonal antibodies. So plasma, in a way, the deployment of plasma clears the way, takes it off the table, and makes it a lot easier to proceed to thousands of people in vaccine studies rather than test it gradually.
What would be the mechanism of ADE in this case when you give someone plasma? What would happen?
I think the mechanism is that it will not neutralize the virus, and it would allow to be taken up, for example, by FC receptors, okay, such that you get more replication internally. The other thing that you worry about is that if you have a lot of antigen and you give antibody, you're going to cross-link FC receptors. Cross-linking FC receptors sets up inflammatory cascades in some ways. So there is, even though, it's very complicated, you have the suppressive receptors, the activation receptors, inhibitory, and the work that Jeff Ravitch and others have worked for the FC receptor. It's a very complex story, but you could imagine that the story could have been very different. And we were all very cognizant of this and being very cautious at the beginning.
So no patient who's received COVID convalescent plasma has ever developed ADE, is that correct?
Well, not that we know. There have been some transfusion reactions. That happens with plasma, but we carried out 2 studies that were published very early in the spring with the first 20,000. And there was, the first 20,000. And what you do is you compare it to the known side effect profile of plasma. And there was no signature. In fact, most plasma is used, surgeons use multiple units. And when they do that, there are often all kinds of problems, like circulatory overload and problems in respiratory. But in this case, they give 1 unit over 6 hours. It was just not there, no signature. There was a DSMB, a data safety monitoring board, looking at all of this, and they never had any concerns.
Now, looking at your NEJM paper, there are a couple of other side effects that I hadn't heard of, trolley and taco. Can you explain what they are?
So those are great acronyms, right? Trolley. So trolley stands for transfusion-related acute lung injury. And this is a phenomenon that you see when you transfuse. A lot of this, that the lungs can be injured, either by antibodies in the plasma or by basically triggering leukocytes to come into it. It's a rare phenomenon, but it can happen. And in particular, it happens if you give many units of plasma. And TACO stands for transfusion-associated circulatory overload. What happens is you have patients that are critically ill, and now you're infusing plasma. And remember, this is giving them essentially a volume into them. And in some cases, that can tip them into circulatory failure. That's why it's given slowly, and that was just not. It didn't seem to be an issue associated with this.
Okay, so just to clarify, these are given intravenously, right?
Correct.
And you say couple of hours?
Yeah, they infuse them. It depends from physician to physician, but in general, these things are infused slowly.
Okay. Now, back to you said that you look for antibodies. So tell us a little bit about that. And does everyone do the same thing? Is that done by the blood bank, first of all? Do they do that?
Yeah, well, last year at this time, there were only research labs that could do this. Mm-hmm. In fact, many places, Sinai, had developed an ELISA. But one of the amazing things that we've seen in the past year has been the very rapid development of these assays. Today, there are dozens of them. The FDA has listed several of them as acceptable for plasma. Mm-hmm. And they are based on your typical ELISA, or some look at optical density. And there are also research-based neutralization assays based on pseudovirus, which you guys know very well about. Okay. Or if you can take it into the BSL-3, then you can do real neutralization.
Mm-hmm. Is anyone better than any other, in your opinion?
No, I would say that the important thing is to do them well, and there is a lot of efforts now going on to coordinate them. That is, a measurement on this one equals a measurement on this one. As you know, serology never gets any respect, but it's one of the most difficult things to study. And antibodies are bifunctional molecules; they exhibit all kinds of properties in assays. But I would say that the science has developed tremendously in the past year. So I would say that we are in good shape in that part, at least today. So
it is not necessary to do a neutralization assay and a BSL-3 to use, in your opinion, to use convalescent serum effectively?
That's correct. Numerous studies have already been done showing the correlation, for example, between an ELISA type of assay or one of the other assays that uses optical density and BSL-3 neutralization capacity and pseudovirus. This is a virus, and this is a system where things are relatively well behaved, and we are fortunate for that.
Okay.
Is there any advantage or investigation into somehow concentrating or purifying immunoglobulin out of plasma to use that instead of just the plasma, or is that just not necessary?
Richard, your question is very timely, because early in the pandemic, many people recoiled at plasma and decided that what we needed to do was to, in fact, make what is known as hyperimmune globulin, in which what you do is you take the antibody and you make a preparation. And those preparations were made, the clinical trials were done, and unfortunately today they reported that they were negative. Now we're waiting to see the data, but it may very well be that they also were treating people who were late. But there is another element to this for the audience who knows about this. It turns out that IgM and IgA are probably the major isotypes that neutralize coronavirus in plasma. There have been several papers about this. And what IgG, by what they do is they take out the IgG and they take out the other isotopes. This is an envelope virus. Complement may be very important in neutralizing this stuff. And IgM is a very powerful activator of complement. So once you begin purifying plasma to make it more consistent, you may in fact lose its power.
Okay, good.
Is there any, aside from COVID-19, is there any other use of convalescent plasma in medicine that we could cite that's going on, in the past years? Or
yes, actually, we are talking about a therapy that was developed in the 1890s. Mm-hmm. Convalescent plasma was used in 1918. Convalescent plasma has been brought out as a defense for humanity in every single pandemic or epidemic. In fact, in this century, it was used on SARS-1. It was used against H1N1 on it. It has been used for MERS. It's been used for Ebola. And they thought of using it for Zika, but the epidemic, it never got to there. But here is the problem. It's always used emergently. It's always used in suboptimal conditions. So one of the things that we want from this pandemic is we want to develop IRB protocols that are off the shelf. So the next time it happens, you could just pull it in and fill in your virus. Mm-hmm. And fill in the assay and do this. And those things will be, you got to test early, you got to use it early. The closer to the beginning of symptoms, the more likely it is to be effective.
So go ahead,
this
Brian.
is antiviral. You guys know this. I mean, what kills what kills people in viral illnesses is often the immune system.
Yeah.
So you've mentioned a couple of times the importance of delivering the convalescent plasma early. So kind of what kind of timing seems to be best here? And how do we find those patients, and how do we actually deliver this convalescent plasma to those patients at that early time point?
So by late summer, the Argentinians have solved the problem. They published in the New England Journal of Medicine a study of elderly patients, who were treated with convalescent plasma within 3 days of developing symptoms. Mm-hmm. And they were able to prevent progression of disease by 60 or 70%. 70% if they use high titer plasma. So that's the answer. The answer is, we now have transfusion facilities for outpatients. We've been using them for monoclonals, mm-hmm. which, as you know, are getting into trouble with the variants. So there are 2 Hopkins trials that are ongoing and should produce data soon on outpatient efficacy. So Brianne, what I would say is, those with elderly, those with pre-existing conditions, once they get symptoms, what you want to do is you want to bring them to a facility and treat them. So basically you're targeting towards high-risk populations. Right, exactly, because they're obviously, the 30-year-old is much less likely to end up intubated, although some do. But, you could also, you probably have a window by which plasma is effective. We don't know that window is, but I will tell you that by the time that people are hypoxic, it's probably not. The window is rapidly closing. Right.
So these are the same.
This issue of earlier versus treating too late. I mean, this has come up with the monoclonals as well, and some of the trials on those, they've had exactly the same types of issues. Also
remdesivir, right?
Right. I mean, any kind of antiviral approach. So in the process of developing these protocols that you're talking about for future use of convalescent plasma next pandemic, one of the advantages often touted for monoclonals is that you can grow up huge fermenter loads of these and process them and produce as many doses as you need potentially. Are you working on protocols that would deal with the infrastructure issue of getting convalescent plasma?
So Alan, first I want to comment on what you said earlier. The results with the monoclonals, the result with plasma, and probably with hyperimmune globulin are consistent because they all have the same active ingredient. I mean, this is a no-brainer. They may differ in amount, but you're talking about differences perhaps in one is polyclonal, the other one is monoclonal. But getting back to your other thing, the infrastructure is already in place in it. With plasma, you need no development. All you need are survivors. And you have a potential therapy. And if we learn how to use this and we can export this information, the rest of the world is going to be dealing with COVID for a long time. And they're not going to have monoclonals and they're not going to have antivirals, but they will have survivors who can then donate plasma. And incidentally, I'll tell you one more thing. Plasma changes. We have a paper that was just submitted today, and it's going to blow your mind. If you want to read it, it's in a preprint server. The efficacy of plasma drops with distance. So, for example, yes. So even though we have been measuring only the titers and neutralizing titers to the S protein, it turns out that local plasma is best. Why? Because local plasma is reflecting the variance in your community. And it's going to blow everybody's mind because the efficacy of plasma by looking at over 30,000 patients drops rapidly with distance, such that 150, your best benefit in mortality is if you use plasma that is local. Colombia just has a very important preprint posted in a double-blinded trial where they showed about a 50% difference in mortality. That plasma came from New York City.
Oh, so you're saying that's why it didn't work so well?
What I'm saying is when you set up a randomized controlled trial, often you want to control all the variables, including stored plasma. Okay. That may not be a smart thing with a rapidly changing RNA virus.
Wait, so you're saying it's best to have local plasma, right?
Local plasma.
Because that's where whatever the virus is circulating, they're right there, right?
Exactly. Okay. And if somebody has variants, then you have variant plasma. In other words, if your monoclonals are defeated by the variants, you know what? As soon as that person recovers, wait 30 days, ask him to donate. You have antibody therapy for that variant.
And which is actually a beautiful piece of information, because you don't need to know that you have variants in order to implement it, right? You don't need to do the sequencing of the virus and say, this is a variant of concern, this, and all that. You just, if you use the local plasma, you benefit from this local effect.
Alan, that data that has just been submitted today dates from the summer. It tells you that during the summer, they were already there. You asked the question, how do variants come to notice? They come to notice because they do something different, right? They are more transmissible. They are more likely to be killed. They defeat a monoclonal. But those, I'm being told by a virology friend that he doesn't like the word quasi-species. So I'm not going to use it. I'm going to call it local variant communities.
I like quasi-species. It's okay with me.
All right. So we are among friends, and the audience doesn't get worked up. So what he's telling you is that this diversity was already there over the summer. This thing was changing, and this is immunological proof of that phenomenon.
So this raises a question for me: is the plasma that's used in any given location, is it pooled?
Never. It's not, Richard. Part of the reason is because you want to be very careful. And the question is, why was convalescent plasma abandoned in the 1950s? Because it led to the discovery of serum hepatitis. And in those days, they used to pool the plasma. It was used, Vincent, for the treatment of polio. It was and preventing polio and serum hepatitis. So by keeping the units separate, you really reduce the risk to the patient. By pulling stuff, one individual, one thing that gets through an assay.
Okay. Arturo, isn't there a situation of people with Gammaglobulinemia? Don't they get, bi-weekly or twice a month serum?
Yes.
Or is that the purified, as you referred to earlier?
They get the purified. They get the IVIG.
Okay.
In that case, but you brought up something very interesting. One of the clearest areas of efficacy with the plasma has been on the immunosuppressed. People who can mount antibody responses, if they're taking rituximab or whatever, they have a bone marrow transplant, and you can see that they basically get a chronic infection. Interesting, they often don't get very sick. Again, potentially because of the immune responses was causing disease. But then the administration of plasma stops the viremia. It's not viremia, the virus detection.
That's interesting because we had Alessandro Sette on last week. I don't know if you know of Alessandro.
Yeah, okay. I like Alessandro, and I like drinking wine with him.
Okay. Well, he said that agammaglobulinic patients don't have any issues with COVID. They do fine, which blew me away because I know that when you immunosuppress with an antibody, that's different. I understand. But people who don't make B cells, they're fine. What do you think about that?
Well, I mean, we have a paper. There have been many papers showing that the individual with B cell defects get into trouble with persistent COVID. And that is a population in which even the FDA has stated that you can treat late. Because in that case, you're really using late treatment, using it as an antiviral.
Right. Okay. So can you had sent this trial, which what happened here at Columbia, where the last author is Ian Lipkin, and this is a double-blinded, randomly controlled trial, right?
Right.
But the
first author is Max O'Donnell,
but they did it in hospitalized patients, right? Which is probably too late, no?
Well, yes. But think about it. There is probably some sort of distribution.
Yeah,
so course. Not every. So people look, when people show up at the emergency room, some can benefit from plasma, some cannot. And if you do enough of them, and if you do high titer plasma, and if you do it early in hospitalization, you may pick up a signal even at that point. And they got a signal. They treated at day 12. Patients have been on day 12, and even from then, they showed a reduction in mortality. Here's an interesting tidbit. their primary endpoint was clinical improvement at day 28.
Right.
But they didn't see that.
Right.
But they saw a reduction in mortality.
Right.
How can that be?
That's what I wanted to ask you.
How can that be?
They didn't get better, but they're not dead, right?
Exactly. What happens is you're looking at 28 days. So if you begin saving people that would have died otherwise, right, and they're still in the hospital, the difference will go away, even though they're still alive. But believe me, I think that everyone in this Zoom call would like to be part of the plasma group.
Yes.
That's that's why I always say to people, look at the curves. What group do you want to be in?
Yeah, right. Well, if it doesn't hurt you, and you've told us it doesn't hurt you, but
it's what I would say is as safe as plasma, and plasma is very safe.
If you had a choice between monoclonals and this, Arturo, what would you do?
If I'm an outpatient in an area where only monoclonals are available, I recommend that people, especially if they have big systems, to get it. But if I, God forbid, get sick and I'm in the ER, I would ask for it in the emergency room.
So that's a good point. There are parts of the U.S. where you can't get plasma, right?
No, plasma is now available throughout the United States. Amazingly, this country has solved the plasma problem. Okay. Monoclonals are more dicey in terms of where you can get them and where they are available, but they're also available in almost all the states now. Although, last week, one of the monoclonals was pulled because it was defeated by the variants.
Mm-hmm. Yeah. You can't use monoclonal therapy anymore. You need to use
cocktails. Yeah.
And of course, your plasma is a wonderful cocktail, right?
Right. So as you begin adding monoclonals to the cocktail, you begin to approximate plasma.
Yeah.
Right.
Of course, you still have very high activity per protein because all your protein is directed towards the virus. In the case of plasma, you have broad activity, but it's low per protein because a lot of it is directed, to everything else, to your tetanus shot.
Arturo, do you know the complexity of the antibodies in plasma? How many different antibodies are present? How many different epitopes are? Has anyone looked at that for
they are beginning to look at that, and there is beginning to be data. People are using arrays. It's a complex protein. We're probably talking about thousands of epitopes. But what I think from a virology, immunology point of view, I always wonder, whether we're looking under the street light. I always said that the best thing that happened to virology and the worst thing that happened to virology was the viral neutralization assay. Why was the best thing? Because you could get quantitative data.
Yeah.
You can get beautiful data. You can write beautiful paper. Why is the worst thing? Because you forget about the FC part of the antibody.
Yeah.
So consequently, what I was going to say is we are focusing on antibody-mediated neutralization. I would love comparable work on ADCC, complement activation, and phagocytosis, because phagocytosis may be just as important. You put in a phagocytic vesicle, this stuff will not go anywhere.
Yeah. So I was actually just thinking as you were talking about that, you mentioned earlier the importance of IgM and complement in destroying this virus. Could one of the benefits of convalescent plasma be that you're also putting in some additional complement proteins?
It's possible, but remember, the volume of plasma relative to the volume that is there is actually very small. It's about, say it's 250 cc's. The volume is at least, I don't remember, but let's say 5 liters. So it's unlikely. What's concentrated on the plasma is antibody. Because your plasma that you're bringing in and the plasma from the patient is probably roughly the same complement concentration. But you may have now antibody at 1/1,000 titer. So you're giving something, that's the agent that is concentrated relative to what they have.
Yeah, I was just thinking maybe that was a benefit compared to monoclonals.
But it may very well be that, we're thinking about viral neutralization, but, ADCC is going to have to be very important, especially when that virus is in tissue. And that's a much harder assay to do. You gotta set up cell cultures, and it's not the same. It's not the plaque.
Yeah.
Is anybody looking at post-vaccination plasma?
Yes. And here is a wonderful story that could help us in the future. It turns out that people who had COVID, when you vaccinate them, they produce whopping amounts of antibody. And by the law of mass action, they have so much antibody that they could even neutralize the variant. Because the reason the variant escaped is just a weaker antibody protein interaction. Mm-hmm. But if you load it up with antibody, even a weak interaction will neutralize. So there is a lot of interest in this because that could give a new weapon, in particular if the variants get out of control.
Well, and also it expands. I mean, obviously the people who had COVID and then got vaccinated get this large response, but there's also a pretty substantial response that we see from most of the vaccines, right? I mean, people who never had COVID but got the vaccine develop a robust response.
Right. That's right. Currently, I believe the FDA is allowing COVID plus vaccination plasma, primarily because it's such high titer.
Right.
It won't be long before that's half of the plasma you can get anyway.
Well,
yeah, right.
Yeah, it's like I said, the great advantage of plasma is it needs no development. It keeps up with the virus, and we just learn to have to use it so that next time we get into this, the first instinct is not to treat intubated patients with it.
Well, and a little ancillary advantage is it's probably a lot cheaper than a lot of the other antivirals and monoclonals that we're throwing at this.
$300 a dose, and you only have to use it once.
How long does it last?
You either get a benefit or you don't. It's just days. I mean, this is and it has no pharmaceutical sponsor, so it doesn't have a bunch of lobbyists trying to push it. It's made from people. It needs no development. If we learn how to use it, we would. My hope is that we learn in this pandemic how to use it so that we can deploy it against a lot of other things.
So in practice, does one obtain convalescent plasma from the blood bank much the same as you would obtain blood?
Yes.
Okay.
Today, yes. A year ago,
that does cut a lot of middlemen out of the business.
Yeah, exactly. I mean, a year ago it was being collected, in specialized places, but the blood banking industry is amazing, and I have got tremendous respect having worked with them. How careful they are, how committed they are, and what I would like is think about it. If we learn how to use this, we may be able to use this for flu, right? Because if you get flu early. You could get donors by December, and you can imagine intervening in a nursing home. Mm-hmm. And every year, you don't have the vaccine. Even if the vaccine is a mismatch, your plasma should match the strains.
So why haven't we done that? Flu's been around a long time.
Because this has just not been, it required an emergency and it required a calamity to really focus people on doing something like this.
Well, and also the attitude toward flu, even in the elderly, is, it's just the flu, I'll get over it. Because, in their lives, they've gotten over dozens of flus, and then, somebody dies of it. And by the time somebody would think of convalescent plasma, the patient is already dead.
It's also interesting. Here is a very interesting sociological aspect. It tends to elicit passion. Some people love it. Some people hate it. Some people cannot believe that in our advanced medicine we are relying on a therapy where every unit is different, where it is poorly characterized, where even if you bleed the patient 2 weeks later, the unit is again different because the immune response is evolving. And other people who basically say to you, no development, cheap antibody, and so. So it's been a very interesting experience.
So how do you counter those ideas, Arturo?
I'm a big optimist, and I love humanity, and I just engage him. And say, but look at this. This is you don't think that way about a blood transfusion for somebody who's anemic or somebody who's been shocked. Think about it from that point of view. Think about the advantages here. This person recovered, and this is what they have in their blood. It could now help somebody. It's cheap.
So as this discussion progresses, I keep thinking about this op-ed, I guess, or short article that you sent us from what was it?
The New
Atlantica?
Atlantis.
Yeah, Victory for Scientific Pragmatism.
Yeah.
Right.
That comments to some extent on the value of, or the relative value of controlled trials versus other experiences in evaluating this. Could you summarize this in your perspective on
So
it?
look, medicine has made tremendous progress. Medicine used to be all about anecdotes. Well, I did this to a patient and all that. And eventually they became an experimental science, began to import ideas from experimental science. Then a few decades ago, it adopted the randomized controlled trial mechanism, which is a very powerful tool. But like all tools, it answers the question that you pose. The problem has become that some people only accept randomized controlled trial data. And for example, the paper published in the New England Journal of Medicine has a dose response. Physicians can't relate to that. Many, I wouldn't say physicians, I said many physicians can't relate to that because they say, but there is no negative control. But I said, it's the active ingredient, it changes. In science, we often rely on that. And what I point out to them is think about other areas like cosmology. Can you do a negative control in space? Can you knock off a galaxy and see if the other ones think? Like a knockout experiment? No. It's a different type of knowledge. And the important thing is that we need to evaluate knowledge from all its sources. You have mechanism of action, viral neutralization, ADCC, complement activation, phagocytosis. You have the fact that you can transfer it to an experimental animal and save the animal. You have clinical observations. You have some randomized controlled trials that are coming in positive. Others are coming in negative. Do you think that if they were giving normal saline, that some of them will be coming in positive and some of them will be coming in negative?
Yeah, probably, right?
So what I basically say is you got to take a holistic view to data. It's what we do. There are fields who cannot do an experiment, and yet we know a lot about the universe. Let me give you an example. The tides on the moon. So we accept that the moon and the tides rise with the moon, right? It took millennia for that association to be made. We accept it today. It matches our everyday existence, but you cannot do the definitive experiment, which is to remove the moon.
Remove the moon.
And see if the tides don't go up and down.
Yeah.
And what I say, so I say to scientists, to physicians, we got to avoid fanaticism everywhere. You need to basically focus on evaluating all pieces of data. And that is you have to maintain an inquisitive mind, a receptive mind.
Well, at the same time, especially in the context of COVID antivirals, I think I would like to point out that you should very carefully apply this idea of whether you should use a randomized controlled trial or not. For something like plasma therapy, it's not appropriate because you don't have a standardized product that you're testing, and so there are all these issues associated with it. For something like hydroxychloroquine,
Yes.
somebody who's a fan of that might say, well, let's go with Arturo Casadevall saying we don't need a randomized clinical trial. But in fact, in that case, you do have a standardized product. You can do a dose response. You can do all of these things. And so you probably should.
Yeah, and it turns out that the trials came in negative.
Yes.
I do wonder, though, I remember early on, listening to it does have antiviral properties in vitro. I do wonder whether it can be made to work. That's another question, right? The trials gave you a negative answer. Are there conditions by which it can be made to work? I don't know that.
Yeah, the reason it didn't is because there's an alternative entry process, right, Arturo?
In this case,
for hydroxychloroquine, so you'd have to inhibit both, which you could do, but the double trial is not continuing, right, Alan? The hydroxychloroquine plus Camastat.
I think that's all been set aside at this point.
Yeah. Because Arturo, the thing is, it depends what cells you use, right? They used kidney cells, which we said in the beginning. Who cares about kidney cells? It's a respiratory infection. Yeah. So use respiratory cells. Yeah, I wanted to bring up the hydroxychloroquine because it was given an EUA without an RCT, right? so someone could say, "Hey, Arturo, so see what happens when you do that."
Absolutely, and that has been the criticism of the plasma. That's been a criticism of the plasma experience in the United States. But I would tell you what's different. We had 120 years of experience with antibody therapies. This had been used in every prior epidemic. There was a high probability that based on historical experience, that this may work. There was a history of safety, and you had nothing else to give. Will you then say you shouldn't give it unless you spend a year doing randomized clinical trials?
So
I would say do the randomized clinical trials at the same time as you make it available. And there has been evidence in other areas in which it's been done. So I strongly support the FDA's decision. I will tell you that paper that I sent to you, the New England Journal paper, this is work that was done primarily by the Mayo Clinic. We spent the entire summer working with the FDA, and I have tremendous respect for them. They were very critical of the work, and only when they saw a dose response did they felt confident to make it an EUA.
How widely used is plasma therapy now in the U.S. and elsewhere?
So I can tell you that plasma therapy peaked in the United States at the beginning of the year when about 30,000 units were being dispensed per week. Think about that, 30,000 per week. Since then, because there have been some negative randomized clinical trials, control trials that have become coming negative, physicians have walked away from it. But not all. For example, in New York City, some hospitals give it to what I would say is today it largely depends on the physicians in the hospital. It's under EUA. The FDA stands by it, and it's available on it. And I can tell you that if I was to get sick, based on what I would know, I would like to get it, and I would like to get it in the emergency room before they send me upstairs as soon as possible.
So you mentioned earlier about infusion centers, so you can get at-risk people earlier. Is that actually being done?
The infusion centers have been set up for monoclonals, Vincent. So they're already there. So if the Hopkins trials was to show efficacy in outpatients, you could just very rapidly deploy to give it to the elderly, in particular as to begin to deal with the variants.
Okay.
So the funny thing is the monoclonal. So plasma helped the monoclonals. The monoclonals led to the setting up of the outpatient infrastructure. By the way, in some countries like Argentina, and I learned this morning in Uruguay, they are beginning to treat elderly patients as outpatients with convalescent plasma already. The clinical trial has been done. I mentioned it earlier. So if that happens and one was to show efficacy, then the existing infrastructure could be rapidly modified for plasma. Mm-hmm. So fingers crossed. Let's hope that they come in positive.
So at this time, there's no shortage of plasma in the U.S. It continues to be collected by blood banks. Is that correct?
They currently the stocks are very large. The United States has collected sufficient, so they are stopping collection simply because there is the amount of it that is in stock doesn't require the outlays. But there is a lot of interest in collecting variant plasma, and there's a lot of interest in collecting some of these. Some people are calling artisanal plasma.
And do you have you kept in touch with what other countries are doing around the world in terms of this?
In fact, I was very involved with that. You know, I'm originally from Cuba, so a lot of contacts in America Latina, and I just got my colleagues in Uruguay want to Zoom with me to talk about what's going on there. Yes, there is plasma is now. What the interesting is, different countries are using it based on their local experience, which is good. Which is very variable, but you know, you have some countries getting very good results, other countries not having very good results. It all depends. I believe that there has to be one answer. I'm a scientist. I believe that all things can be reconciled if you understand the variables. And I believe it's negative results are relatively easy to get. Positive results are much harder to get, and this is why we value them more. And if you begin to get positive results from multiple areas, that's probably a signal that you can believe.
So you mentioned this relationship with distance. Is there much of a relationship with timing of collection of the plasma? Can you collect some plasma and store it for a while for a future outbreak or something like that? Or does it need to be used right away?
So, Brianna, that's a very insightful comment. I think that before we learned this, we thought that you could collect it and store it for a long time. You can probably do that with a virus that doesn't change very much. But with a rapidly changing RNA viruses, we're learning with this is that you're taking the risk that, especially if you're measuring only one thing like viral neutralization, it turns out that maybe the important thing is the binding of the other epitopes that lead to ADCC or complement activation. You know, it may lose efficacy. This is why I think we need a lot of very basic science to be done on this to sort it out. The clinical data has gotten ahead of the basic science.
You know, I want to riff on something you said earlier. You were lamenting that the neutralization assay made us focus on ignore the FC, okay? I would argue it made us ignore T cells,
Yes.
Yeah.
which apparently, according to Alessandro, are going to save us. And it's just too bad you can't give T cells, convalescent T cells, right? Because then you have other matching issues, right?
Well, there may be. Maybe those therapies can be developed. I mean, I'm a big optimist. This may just be the first layer of defense.
So let me ask you one more thing, Arturo. What do you see the role of convalescent plasma for the remainder of this pandemic, for however long it goes?
So if you had interviewed me a year ago when we were setting up, I would have said to you, Vincent, this is going to be a few months. Monoclonals are coming, antivirals are coming. Plasma is a stopgap measure. We just try to help some people until we get to a higher ground. A year later, plasma is still standing. We're beginning to see problems with other. I think that unfortunately, but the unfortunate is because we don't have better options, we are going to be relying on plasma for a while. And my hope is that this enormous effort galvanizes people to look at other uses of it, so that then we can not only use it for other diseases, but also with the next virus, which whatever hits us, that from day one, that we could, quote unquote, do the right thing.
Arturo Casadevall, Johns Hopkins University. Thanks so much. Pleasure, Arturo.
Honored to be interviewed in virology.
Great to talk to you. Thanks.
Thank you so much.
You work with things you can't see. I work with. Anyway, thank you, guys. Bye-bye.
Bye-bye.
See ya.
Take care.
Well, I must say I have newfound respect for convalescent plasma.
Yeah.
Because, Daniel is not a fan, right? He's a big monoclonal fan.
Daniel is a fan of random controlled trials.
He is, yes. And I think the good case was made today why you can't always have them. And the Atlantic, what is it called? The Atlantic?
The New Atlantis.
The New Atlantis.
Okay. That was a good article on the issues, right? We'll link to it and see. I don't know if it's under a paywall, but I was able to get it.
No, I was able to get it.
No, I think it's not.
New Atlantis. In an emergency, the gold standard of evidence may not be the wise one. I mean, that's the bottom line, right? It was an emergency. We tried to do something.
And I like his...
but there is this fine line you have to walk so that you don't start dosing everybody up with drugs that don't work, which also happened early in the pandemic based on, this patient got better. So I appreciated his distinction there that, if you can do a randomized controlled trial, you really ought to. But if you can't, as in the case of plasma therapy, and really most therapies are not like convalescent plasma therapy, right? Most of these are more standardized products.
Yeah, that's the key. It's not a standardized product.
It's not a standardized product. And I think that, as he pointed out repeatedly, that's one of the real advantages, is that it's almost like sort of homegrown personalized medicine.
Yes.
All right.
Small batch artisanal medicine.
That's right.
Artisanal plasma. And look, it's got a history, and they've shown it's safe. It's not going to hurt you. So, those are the supporting points.
And there's existing infrastructure, and it's cheap.
Yeah.
was cheaper than a monoclonal. Now, yeah, it's a tough one. It's a tough one. Yeah. If someone said, I don't have any monoclonals, you want plasma? I'd say, yeah, sure.
Sure.
But I don't know if they gave me a choice.
Can I have both?
Yeah.
You could. I don't see why not. It's a lot of money. Okay. That was good.
Yeah, that was good.
A few announcements before we move on. Don't forget NIDO 2021. It's the online symposium for coronaviruses and more, happening June 7th through 8th. A lot of good speakers. I've registered. It's going to be a Zoom type thingy, and check it out. We'll put the link to that online. Vaccine education town halls, Rich.
Yeah, just to replug this for people with questions about vaccines, COVID vaccines, or vaccines in general, how they work, what the issues might be, concerns, whatever. American Society for Virology, in collaboration with American Society for Microbiology and the University of Virginia, has put together a series of Zoom meetings. So you can find these at asv.org/education. And these are ongoing. You can click on register and you'll see a menu of different meetings, each hosted by at least 2 out of a pool of 50 experts. And you can just jump onto one of these and for 45 minutes or so, ask these people your questions. And if you don't get all your answers, sign up for another one. And if you don't get all your answers, sign up for another one. Come back as many times as you want. I would also say that we've also put together a sort of a sample / promotional video for this that's now done and released. You can find it on the ASV site. It lists to a couple of videos. One is just like a 30-second trailer, and the other is this 6-minute thing that gives a bunch of sample frequently asked questions and shows some of the experts giving their spin on it. And we can also put in a link to the YouTube archive of that same thing. So join us. This is a resource if you got questions.
Yeah. A lot of fun.
Yeah.
Kathy Spindler has a research lab specialist intermediate position, microbiology and immunology, University of Michigan Medical School. They're recruiting a highly motivated individual for a full-time lab research position with substantial research and lab management responsibilities. Lab performs fundamental research in viral pathogenesis of mouse adenovirus type 1. Master's or BS plus 3 years of experience. Preferred position open immediately. Comes with excellent benefits. We will put the link in the show notes. You can also find it on the ASV job sites, which you can find at asv.org. All right. One more little brevia, which I thought we should touch on. Daniel talked about this, but I think it's worth repeating here because we have different listeners. A press release from Pfizer BioNTech. They have good results from their vaccine study in adolescents.
There has been much rejoicing in my household about this.
Yes. You have adolescent? You have an adolescent?
I have an adolescent. She's 15. She wouldn't be able to get this vaccine until the beginning, sometime into the beginning of next school year. And I said, well, they're doing a trial. They're 12 to 15, and I'm expecting the results sometime soon. And so they came out, and I sent the link to her, and she's very excited. They
enrolled 2,260 adolescents 12 to 15 years of age. This is in the U.S. 18 cases of COVID were observed in the placebo, none in the vaccinated group. Good antibody titers induced 1 month after the second dose. And that is, what is the percentage there? 100% of efficacy against
because they had no cases
disease.
in the vaccine.
No cases. And very good antibody responses. So that's all good. And Daniel said yesterday, the goal is to get everybody 12 and up in the U.S. vaccinated by the end of June.
Yep.
That would be cool.
Yeah.
So I have a couple of comments. First, they don't, in this press release, it's a press release, so we'll have to wait to see the paper. But I'm getting more and more comfortable from that with that.
They have to do this because everybody who knew about it would be trading stock on
Right.
this basis. Yeah.
They do not say what their endpoint is for efficacy. I assume that they're scoring
COVID, right?
but symptomatic, right?
Yeah,
They're
sure.
swabbing all these kids.
No, probably not.
So it's they're looking for symptomatic disease, and I would point out that in terms of claiming 100% efficacy, although I don't know what the confidence interval is on this because we're only talking about 18 cases.
Yep.
In the larger trials, the initial trials, where you're talking about 30 or 40,000 individuals, they made statements about that, drew conclusions about that when they had 150 or so cases. So this is many fewer cases, but I imagine that given the experience so far and our understanding of the correlation between an antibody response and protection against disease, that they, I assume, I don't know, that they aren't going to be looking for a full-blown trial. That, well, obviously they aren't. This is what do they call this in terms of phase? Is this a...
This is a, oh gosh, what is
They
this?
don't
It is a phase 3.
Phase 3, yeah.
But it's because this is an already approved vaccine in another group. The standards for it are not nearly as high.
Right.
And not only is it an already approved vaccine, it's now been given to millions of people.
Right.
And we've got very clear data on everything from efficacy against disease to now efficacy against infection. The original group has been followed for more than 6 months, and we know immunity is robust. And so we've got all of these correlates of immunity that we can measure and say, okay, in adults, this is how it looks.
Right.
And this trial is the first step in the direction of getting it for everybody, down to 6 months old. And you're looking at an age group 12 to 15. They're very, very similar to adults immunologically. I mean, there's not a lot of immunological weirdness going on the way you would get with, say, newborns or even kids. These are adolescents. They're mostly matured. And so you're looking for those correlates. You're looking for the antibody response. You're looking also at the efficacy. Did they get sick? And this all lines up. And I think it's assuming that the data match the press release, which based on Pfizer's history with this, I think we can probably be pretty confident in. This ought to be enough for the emergency use approval.
Go ahead, Brian.
They have a section in this press release where they talk about the other 3 trials they have underway. Mm-hmm. One in a group of children who are 5 to 11, one in the 2 to 5 year old group, and one 6 months to 2 years old.
Yeah.
Yep. And they refer to those as part of a seamless phase 1, 2, 3 study.
Yep.
Yes.
So is that the ongoing, or is it a new 1, 2, 3?
So they said that they dosed their first children in a seamless phase 1, 2, 3 last week.
So is that part of the previous 1, 2, 3, or is it it must be part of the previous 1, 2, 3, because that was seamless also, I think. The one that culminated with the approval of the mRNA vaccine. So I think this is part of that, right?
This is right. So this is a separate study because it's in a different age group. But it's the same structure.
I know, but so you're saying it's a separate 1, 2, 3 study, right?
Yeah.
Right. In children 6 months to 11 years old.
And whereas the 12 to 15 was a phase 3 immediately because you say they're like adults, right?
Right.
Immunologically, at least.
Yeah, they started. They started the 12 to 15 group right around the time, right after they got the emergency use approval for adults.
Yeah. But that was a phase 3. That was already a phase 3.
And they said, okay, let's do a phase 3 in the group that we're pretty sure this is going to be the same in.
Okay.
And now, based on that, they're starting this 1, 2, 3. When you get down into and I'm sure Brianne can talk more about this. When you get down to the 6 months to 11 years old, that is a huge range of physiological and immunological changes. I mean, or as any parent would also know, you're going from diapers to puberty here, and there's a lot that you worry about in that, and so they've got to do the phase 1 and 2 stuff that they would do in a full trial there.
Okay.
One of the things they say here is we plan to submit these data. Ooh, these data. Look at that.
Yes.
To FDA as a proposed amendment.
Yes.
To our emergency use authorization in coming weeks and to other regulators around the world, with the hope of starting to vaccinate this age group before the start of the next school year.
Yeah.
So there's a couple of things there. They're giving a sort of a timing, and I don't know whether the how the review process for an amendment to the original EUA, the review process is probably pretty similar.
It's pretty similar.
I would imagine, but it's not a whole new EUA. It doesn't need to be. It's an amendment to the EUA.
Right.
Okay.
And I don't have all the dates and times on this, but the other companies that are developing these vaccines and that have approvals are also doing pediatric trials. J&J immediately went, being Johnson & Johnson, of course, they immediately went for the pediatric trials after getting the adult approval. And theirs, I believe, is a 6 months to 17 years trial. So they're just doing the whole spectrum. And I think that's a phase 1, 2, 3 type of structure. Moderna is taking a little bit of a different tack. They saw that Pfizer was doing the 12 to 15 age group. So I believe they've already started the 6 months to 11 years old. And then they're subsequently maybe going to get around to this 12 to 15. But I think they're kind of thinking, well, Pfizer's ahead of us on that. We're just going to turf that to them. And the Moderna vaccine, which is highly similar, might come out sooner for the younger age groups.
That makes sense.
I am so impressed at these data. Holy cow.
Yeah, look at that.
Holy cow. People
Good usage.
Somewhere know the right way to do it and don't argue with us that who cares, blah, blah, blah.
They're all listening to TWiV.
Thank you, Pfizer marketing
just-
people who put together this press release. We appreciate your
great.
use of these data.
I like that. And by the way, Daniel yesterday was talking about, we're at a plateau in New York. The cases are not going down. They're going up a little. And he said, it's not a variant of concern. It's behavior of concern. Yeah. It's people.
Yeah. We've got this going on in Massachusetts right now, too. We've got our 7-day average went down, it plummeted and then plateaued, and it is now coming up. And the politicians are going on about, we have to worry about the new variants. And I'm thinking, you have to worry about the fact you reopened all the damn restaurants a month ago. Right. That's why you've got this happening.
Oh, but Alan, it's so much easier to blame.
It's so much easier to blame the virus. Yes.
And that's an unfortunate message.
Yes.
It is.
Because people are likely to think, oh, well, there's variants. I can't do anything about it.
Exactly. It's going to exacerbate the COVID fatigue. This whole attitude of, we've been tolerating this for a year. We're done. It had been explained as, we got to keep doing this for another few weeks, maybe a month, while we get enough people vaccinated. Just hang in there. We're almost done. I think that would have been a much easier pitch. But now, the genie's out of the bottle. We're in reopening, and yeah,
air travel is going up.
Yeah.
All right, let's do some email. I think, Rich, you ought to take that first one.
Okay. Kareem writes, hello, Dr. Racaniello. As a pandemic-inspired listener, I wanted to thank you and your team for the thoughtful and interesting discussions. TWiV has become my favorite podcast, surpassing Mark Marone's WTF podcast. As you may know, Peloton has evolved into educational and inspirational workout sessions. As an example, there's a spin bike instructor who gets people moving while telling an entertaining story about Oprah's interview methods. I look forward to a future Peloton session with Dr. Condit summarizing a research paper while enthusiastically shouting out spin bike resistance levels. All the best, Kareem. Well, I'm for sale. So if Peloton wants me to do that, I'm all for it.
Yeah, but people would accuse you of spinning the paper. Also, you're a rower, right? Shouldn't you be doing this on an erg?
Yeah. Well, yeah, I suppose.
Yeah.
I can see you doing that. Yeah.
Yeah.
You got the
Why
voice.
not?
You got the. Do you have a Peloton?
No.
Well, no, they'd have to provide him with one.
Yeah,
I don't either.
that's right.
I don't need.
Actually, I much prefer things that actually go somewhere.
Yeah.
Okay. Or me going somewhere. That's why I much prefer an actual boat to an erg. Ergs bore me.
I agree. But I don't have a Peloton, but supposedly you can, do video and go all over.
Yeah,
the
that's
place
the whole idea.
Blah,
Yeah,
blah,
go all
blah.
over the place.
Oh well. Brienne,
I could be shouting on the screen while we're touring a lab. People pipetting and stuff. That'd be good.
all right, level 10. Let's go. Could see him doing that. All right, Brienne, can you take the next one?
Sure. Krista writes, "Hey there, TWiV team. I've been listening since the beginning of COVID and am sincerely thankful for your insights. I'm a stay-at-home mom with 4 kids ages 17 to 4. You guys really helped me make good decisions to keep my family safe. I began volunteering at the mass vaccination site where they could administer 2,000 shots a day. Because I volunteer, I was able to get a leftover dose fairly early on, and now I'm fully vaccinated. I also donate blood regularly to the Red Cross. It was interesting to see the way that they gave antibody test results once I was fully vaccinated. The whole time I would come back negative. However, after my last donation, I came back as reactive. For a second, I worried that I had contracted COVID, but upon reading the results, I see that this is a new step that the Red Cross has added to verify that a person has been vaccinated, the vaccine is working, and they remain negative for COVID antibodies. So cool. I donate again in May and find these little bits of feedback fascinating. If my vaccine wears off, I would actually be able to catch it, right? Mm-hmm. Below, I will link the screenshots of the Red Cross results so that you can see the way it's presented. I do have one question, and it's probably dumb and not related to the Red Cross thing at all. Question: at what point in a virus's evolution does it become a new disease? We have SARS-1, MERS, common coronavirus colds, and SARS-CoV-2. Can they evolve to a point where it's considered a new virus, or is evolution off of one of the identified virus always a variant of that virus? I'm sure my terminology isn't perfect. I mostly talk to kids and chickens. Anyway, thanks for what you do. Stay cool, Krista.
What do you tell the chickens, Krista?
Tell them, stay cool. Hello, chicken.
So that's interesting. So the terminology reactive is to distinguish from positive, right?
Mm-hmm.
Cool. That's interesting. It's an interesting thought. And there, she sends a screenshot of her reactive result. Yeah. So they must be looking for I don't know. What are they looking for? Because it's a spike vaccine. They must be looking for spike antibodies, right?
Yeah. So they're looking for spike, and I assume the backup test is N or something.
N or something.
Probably,
like
maybe
that.
they do both at the same time to make sure you're just vaccinated and not infected, right?
So there's some stuff posted in the show notes. I think this is maybe from Kathy.
Yeah, this is from Kathy.
And it indicates that the Red Cross is using 2 antibody tests, both spike, one for spike and one for nucleocapsid.
So they do the spike test on everybody, and if your spike comes back positive, then they do the other test, I think, is what this is indicating.
Which we just said, right? Yep.
Yeah.
So when do we have a new disease? My first reaction to that is that one of the firmest distinctions we make between closely related viruses is whether or not they're serologically cross-reactive. So that if they are not serologically cross-reactive, if you get the disease to one, you are not immune to getting the disease with the other. Okay. Whether that means the disease symptomology could be very similar. The disease could be very much the same. But to me, if you're talking about variation that is sufficient to classify something as new, I think I would use that cross-reactivity as probably one of the best empirical definitions.
I would call that a new strain. at least.
Okay.
Maybe not. I mean, because different strains of flu would not be different.
Yeah, but
viruses.
that's right, because it's still influenza.
Right.
Yeah.
It's still influenza virus disease.
I'm sort of under the impression that there's a specific kind of genetic similarity bar that would be important for finding a different virus versus a different strain, though I don't have a number for you off the top of my head.
The strain is easy. That's a It's a variant with a distinct biological property. And as Jens Kuhn has written, given the name strain is done only by an international committee of experts. So that's what a strain is. You can't just say this is a new strain.
Right.
Even if you find it in your lab, it has to be agreed on.
Right. But then there's another bar to be a new virus or a new species.
Right.
So that's a good question. This is the way I look at that. So the examples that Krista gave, SARS-1, MERS, common corona, SARS-CoV-2, these are all independent spillovers from animal reservoirs. And they're different, slightly different viruses to begin with, or more than slightly in the case of SARS-1, MERS, and 2, and they cause different diseases. And so to get a new disease, you have a new spillover. We have Nipah virus, we have Ebola virus, etc. The question is, does a virus ever change in the course of its circulation to become a different disease? And I can't think of one in people, but I can certainly think of non-human animal virus diseases where the patterns of infection have changed. And coronaviruses in pigs, for example, have done that. But, smallpox for all the years caused a similar disease, and influenza in humans, similar disease. So I think once it's in people, for some reason it's not changing. In the animals, it's a different story. But if you want a brand new disease, just get a new spillover.
I think it's a question of time scale.
What do you mean?
All right. So smallpox has not changed over the historical period in which we've looked at it. That's
all we have, right? That's all we have.
Right. That's what we have. Exactly.
Yeah.
So it came from somewhere, probably a spillover event. We have pox viruses in nature that are candidates for that. But if you look at something like enteroviruses, I mean, are all of those independent spillovers, or could some of them have evolved from others that were already circulating in humans?
I don't know.
Yeah. See, I wonder, I was thinking about Ebola with this. If we think about different outbreaks, we often talk about those as being related to different spillover events.
They are.
But we still all call those viruses the same Ebola virus. Sort of, we put those in the same species, Ebola virus.
Yep.
And so I guess maybe I'm taking Krista's question in a different way. So maybe I just read this wrong.
No, she wants to know when a virus, can be considered a new virus.
So I'm thinking more like where do we draw the line between all of those Ebola viruses versus Marburg?
Right.
Well, that's done genetically, right? You do this genome sequence and you put them into different genera, and same thing with coronaviruses, but it just so happens that those Ebola viruses cause all the similar disease, right? But maybe the genetic difference is not as great as the genetic difference between SARS-1 and 2, which are causing slightly different diseases, right?
I would say, Krista, that if you can ask a question that has 4 scientists discussing it for 10 minutes, it's not a dumb question.
Not a dumb question at all.
No.
And not only discussing it, but failing to answer it.
But, the talking to chickens, I think, has done you good.
Yeah.
And I think the bottom line is there's no textbook definition for this.
Well,
but these are some of the ideas.
And part of the problem, too, is that microbes in general make a hash of our whole notion of species. I mean, we say this virus is this virus is that virus, but there's a whole spectrum of recombination events and changes and such that can go on there. And then, how much of this is just us categorizing things for our own sake?
Right. Well, it all is. It
all
is,
is,
right?
yeah.
It's just us. Get rid of the humans and everything is sorted out.
That's right. Then it all makes sense.
But one more thing on this point, the prevailing idea among coronavirologists, and we have absorbed this by listening to them, the common cold coronas originated by spillovers years ago, and probably they caused severe disease. And now they don't. Of course, that's an hypothesis. So that's one idea for how the way a virus causes disease has changed. So, and as Alan said, we're limited by what we know, and we don't know. We can't go back very far for viruses. By the way, someone wrote in, and she said the best part of some TWiV, which I don't remember, was listening to 6 scientists try and figure out how RNA sticks around for a long time.
Yes.
Well, I think that's what people like when we try and figure out shit. All right, I'll cut it out. All right, so that was Brianne. Alan, can you take the next one?
Sure. Spencer writes, I'm a frontline internist diagnosing and caring for patients with COVID-19. I'm also a graduate of Cornell University Mount Sinai CUNY Graduate School. My question deals with the replication competency of the COVID-19 virus. The CDC instructions are that an infected patient can end quarantine after 10 days because the virus is less capable of replicating, although still could be found in nasal swabs from the patient by PCR amplification. Is it because the host is not likely to replicate the virus and the immune response decreases the infectivity? Why is this? I understood that viral replication assays show decreased replication after 10 days. Why does the virus lose its ability to replicate in a new host? Are there any markers that might be used clinically besides a viral replication assay to see if the virus has been cleared or has reduced transmissibility in the host? And importantly, also not likely to be replication competent in a new host? Thanks. I've been listening to TWiV and TWIP since the very beginning. And Spencer is an MD PhD. So I think that the issue here is maybe in the CDC instructions, they phrased this poorly if they said less capable of replicating, because I don't think we have any evidence, or I would find it very surprising if the virus had become less capable of replicating. What's happening is with the immune response, it's stopping the virus from getting out until eventually there's no virus left. The virus that is in a cell is going to replicate in the way it normally replicates, and it's going to follow through its life cycle in the way it normally follows through its life cycle. So there's nothing changed about the virus's ability to replicate. It's that the immune response is now killing off the cells that are infected and is now producing antibodies that bind to the virus in the bloodstream and is, expunging it from the host. So that's what's changed, and that's how you get the titers coming down. It's not a replication issue.
Yeah, I think I would point out that to Spencer that when we're doing PCR, we're measuring viral nucleic acid, not the actual virus itself. And so we're saying viral nucleic acid that might be sticking around later on is less likely to be from actual virus. It's more likely to be debris.
Right.
Betsy, you have the same pedigree as I do. Cornell, Mount Sinai, CUNY. Great question, Spencer.
Yes.
Yeah, Spencer's probably not the only person who has been confused by this.
Yeah.
So it's good to bring it up and sort it out.
All right, Tracy writes, I've written to you guys a couple of times already, but to no avail. I appreciate that you must get a ton, T-O-N-N-E, of mail. So no hard feelings, but I thought I'd keep trying. You should keep trying because... yeah, we get a lot, and never, ever, ever, ever give up. You know who said that?
Winston Churchill.
He did. Don't give up. There's no point in giving up.
I think Winston Churchill was even more emphatic than that.
More evers?
Yeah.
And I was just going to say, does this mean that I watch too much college basketball that I was going to say that Jim Valvano said that?
Yeah, but he was quoting Winston.
It sounds like... yeah. Yeah, Winston said it. And it's true. I think sometimes you don't try hard enough. But I know, Tracy, you did write a lot of emails. I'm sorry I missed them all. But
by the way, as long as we're here it is.
Here it is.
This is the lesson. Never give in. Never give in. Never, 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 never. In nothing, great or small, large or petty, never give in except to convictions of honor and good sense. Thanks, Winston.
A lot of people don't like Winston, but I think the quotes are fine, Yeah, so thank you, Tracy. She wrote a more recent email where she was very sad. And she said, what do I do? And very few people do that, right? What do? I really want to, your answer. So I felt badly. And so that's appealing to our humanity. That always works. Anyway, I'm writing to you once again from Noumea in New Caledonia, where we have had a crazy few days. A tsunami warning on Friday, followed by Category 5 tropical cyclone Niran on Saturday, and the cleanup operation on Sunday, marred by the announcement that SARS-CoV-2 has now broken into the community here. Oh, I wonder if it used wrenches or hammers.
They're all safe crackers.
They're all safe crackers. That was a great title. It had been contained since we'd first had a successful 4-week lockdown last March, which was followed up with a very strict government-controlled quarantine. Obviously, something did not work, and we are now back in lockdown from midnight tonight, 9th March. There are rumors aplenty bouncing around about patient zero, but these are not helpful. What we need is for the French government to up the vaccination program here. Only 5,210 people have received at least one jab to date. And for all of us who have hitherto enjoyed a pretty normal life, to get with the global program and change our behavior, which is certainly not pandemically prepared. Is that a phrase?
It is now.
Sounds like it to me. Ditch the bisou, stick a mask on your face, and keep your distance. Seems obvious to those who have been doing this for a year already, but we are woefully behind here. I wish that TWiV were also available in French so I could share your advice more easily, but I guess there isn't a French equivalent. No, je suis désolé. Non existe pas. I'm really sorry. N'existe pas is probably the right way. See, I can't even speak French. But, you know, you could get it translated. Well, maybe not the vocal. Yeah, not yet.
Yeah.
But there are programs that you can feed words into and they'll translate it, right? Like,
Sure, yeah.
Right.
Google Translate. Well, that's
Because not always the best.
But
Right.
there are dictation programs
yeah,
where you can.
right.
So anyway, as vaccine hesitancy here looks likely to be a problem, is there any advice you can give me on how to convince those who are a bit hesitant to get involved once we have a better supply, of course? I know you've posted a lot of useful links, but if you know anything in French, then that would be one less hurdle for people. Thank you for what you do. It's truly wonderful. I arrived at TWiV last year through Immune when I was searching for good info on vaccine development. Your podcasts are so addictive and motivate me when I'm cleaning the house. I'm with you for the long run, I hope, as I am only in my 40s. Tracy, Welsh person living in New Caledonia. Well, I know we have French listeners, so let us know if there are any good links to pro-vaccine things in French that we could send along. But as Alan has said before, frankly, if you make it personal, you know, I had my whole family immunized and they're all good and I wouldn't have it any other way. I think if you say, because many people ask us, are you taking the vaccine? As if we might not. And I think that's one way to do it.
The other important thing that Paul Offit and others were really good at this is to acknowledge people's concerns. Okay,
Yes.
when people are concerned and they ask you a question, you listen to them, okay, and say, I get it, you know. Paul Offit always starts out, he says, of course, you know, if you're going to undergo some sort of medical treatment, you ought to ask questions, okay? You shouldn't just take whatever random thing somebody comes at you with, okay? So I'll listen to your questions and I'll give you the best answers I can.
Yeah, I always have been trying to remember that as I've been communicating with people about vaccines, that, you know, sometimes it feels like they're asking me these questions because they want to, you know, annoy me, knowing how much I like vaccines, but they're not. They're asking because they have legitimate questions. And sometimes their question is that, you know, because they want to protect their kid. And that's actually a value that they and I share. And so if I think about it as, hey, look at this value that we share, let's talk about how I think the data says that the vaccine actually is better for that. That's going to be much more useful than me kind of talking down to them. But I have to remember they're not doing it to make me mad.
Right. Unfortunately, these interactions are tend to go a lot better in person than they do online. So if you're interacting with somebody through Facebook or Twitter, first of all, there's a reasonable chance that they are doing it just to annoy you. But secondly, you know, the format of it is such that it encourages extreme responses and outrage and such, and that nobody would engage in a normal conversation. Now, under the conditions of the pandemic, of course, we're not supposed to have in-person conversations because then you're spreading the virus. But if you can, if you're there in the community, and I assume New Caledonia and Noumea in particular as a fairly isolated, cohesive place, there are local interactions you could have maybe through a window, or if you can have Zoom discussions with people, that would be a good format for that sort of thing.
But sometimes, Brianne, they do want to annoy you.
Yeah, I know.
All right, let's do some email.
No, we did email.
We did email.
You want to do pics?
Yeah, we could do that. Brianne, what do you have for us?
So I have a New York Times article that I read recently. I thought was pretty cool. It's called "The Ocean's Youngest Monsters Are Ready for Glamour Shots." And this is about something called black water photography, where people are diving into deep parts of the ocean and taking photographs of larvae of a number of different types of fish. So this talks about this idea of black water photography and people who do it, but it also shows a lot of photographs of some of these. And these are some really beautiful photographs and just intriguing animals. And when I think about, some of the things we teach the students as biology professors, we're using preserved animals and things. And this talks a lot about how much you can see and learn from these delicate creatures that you don't get from a preserved specimen, and all of these cool things you can see. And I just love the pictures in this article. They're so cool.
They're terrific.
This is cool.
Dixon will like this.
Yes,
I did think of
like
Dixon.
this.
Yes.
Yeah,
Actually,
I really like the lionfish here at the top. He's spectacular.
I really like the lionfish. At the bottom, there is a hairy goosefish.
Goosefish, yes.
That one's pretty cool too.
A few years ago, I remember hearing about this this deep, awful offshore night dive, black water dive off Kona, and thinking, gosh, that sounds terrifying, and I want to do it.
Yeah.
Cool.
very pretty. Ellen, what do you have for us?
Well, last week I recall picking something that was kind of a downer for everybody. So this week I'm picking something that should be a lot more fun. This is a game, but before you run away and say I'm not into video games, this is it's not a twitchy reflex thing or anything. It's a series of puzzles, and it's a tremendous amount of fun. It's a game called 7 Billion Humans. This is available on pretty much anything that has a screen. So it's on Windows, Mac, Linux, Nintendo Switch, iPad, iPhone, whatever. However you're listening to this podcast, you can probably play this game called 7 Billion Humans. And the theme of it is you're in an office building, and you are required to give instructions to the office workers to accomplish various tasks. And you do this procedurally, like you're writing a computer program, right? And you have to come up with a procedure that they will literally follow. All of them will follow your instructions to accomplish the given task. Move this box over there, but you've got to get them moving the right space. And all of the puzzles are built around that concept. Now, the stealthy part of this is you're actually learning fundamental concepts in computer programming, but don't think about that. It's just a fun game. So it's a real blast.
I'll give it a try.
This looks cool. I want to try it.
Yeah.
Yeah,
I played it on the iPad, so that's a convenient format.
Yeah, Brianne, take a break from basketball. Try this.
Yes.
Sounds good.
Rich, what do you have for us?
Sort of almost in line with Brianne's pick, and I too was thinking about Dixon when I picked this one. This is 15 fascinating animal x-rays from the Oregon Zoo. And interestingly, the publisher here is something called My Modern Met, which by itself is interesting. Looks like an online publication, I assume, from the Metropolitan Museum of Art, though that organization is not credited anywhere, even in the about information. But it looks like a very interesting online publication. At any rate, this is one publication of that. The Oregon Zoo's in Portland, and this was done by, I guess, veterinarians from the zoo. They get a lot of information about their animals from x-rays. And so they've published a collection of these x-rays. We got a python. We got a beaver, a beaver's tail. Mm-hmm. We got a tiger's paw. I
like chameleon. The flamingo.
Yeah, the flamingo. Great stuff.
It's cool.
They're beautiful pictures.
This is awesome.
These are cool.
You think these are just things they had in their files and they pulled them out?
I suppose something like that. I don't know. But I'm always fascinated by how these wild variations in form are all based on the same fundamental design.
Yeah.
You know, I'm sure I don't know that how true that is in the extreme. For example, snakes, I think, have more vertebrae than me. Okay.
They do. They do.
But yes, endless forms most beautiful. They are.
Yes.
That's cool. Very nice. All right. My pick is a website from BioFire. BioFire makes clinical lab tests. Biofilm is this new way of doing syndromic testing, where they have different panels where if a patient has what looks like a respiratory infection, you put a specimen on this respiratory panel, which is basically a nucleic acid test. Turns around really quickly. You can have blood panels. They have GI panels, meningitis, encephalitis, pneumonia panels. Very cool. Anyway, I was at Emory a couple of years ago. I was having breakfast, and a guy came over, and he said, "Are you Vincent Racaniello?" And I said, "Yeah." And he said, "I recognize your voice, and I work for BioFire, and we upload all the data we get from these films, and you can look at it." And he gave me the website. I thought that was cool. And just recently, Amy sent me this again. I'm picking the respiratory pathogen trends, which is a map of what they've found. And it goes from last 2019 April through, roughly the present. Adenovirus, coronaviruses, rhinoviruses. We're interested because rhinoviruses are kind of popping up again. They have been since July. But you could look at influenza, and you could just focus on influenza. You click on influenza, and you can see, there was a big spike at the end of 2020, beginning of 2021, and then, beginning of 2020. And now this year, there's nothing, just a little blip, 0.5%, whereas at the peak last year, it was like 11% of all their specimens. So they got bacteria in there too. Of course, coronaviruses. It's really cool.
Yeah, I really like how you can specify. So you can, pick coronavirus, and then you can specifically look at either 229E or HKU1 or NL63 or OC43. It doesn't have SARS-CoV-2 in it. Because I was kind of looking for that signature. But I realized I could actually pick individual coronaviruses, and that was cooler.
Let's do that. Let's turn off, let's just do coronavirus, and then we're going to pick 229E. Mm-hmm. So that's yellow. So it's not much. Let's see about HKU. HKU is more. There's a lot more.
And a much more distinctive peak for HKU, whereas 229E is more blippy.
And NL63 is kind of comparable. And let's see, old cow 43. That is kind of reddish. Yeah, it's also not bad.
And it kind of looks like there's NL63 around this year.
Yep.
Yeah.
Yep.
Look at RSV, respiratory syncytial virus.
Okay. Yep.
It's like gone.
It's gone.
Isn't
but
that
last year it was
amazing?
like 15, 20%. Whoa. Yeah. Yeah, it's gone.
Well, in influenza, this is one that's already the CDC has noticed.
It's gone.
It's just gone.
However, if you look at rhinoviruses, they are rhinoentero. They group them together. Let's just have rhinoentero, please. Get rid of everybody else. I want to, I don't want bacteria. I don't want influenza. I don't want respiratory. So rhinovirus, no, that's influenza. I don't want that. Come on. I want rhino. Okay, so rhinoentero is big time.
Yeah. I wonder how much of the difference has to do with the mechanism of transmission.
Yes. I was just thinking that.
I mean, maybe masks are good at preventing RSV, are better at preventing RSV, for example, than they are in preventing rhino. Because rhinovirus is, right, that's shaking hands, right?
Probably that's a big component.
It's a fomite thing.
Yeah, I mean, at least with the viruses, do we see kind of a different pattern with the enveloped and the non-enveloped?
So rhinovirus, non-enveloped.
Let's look at adenovirus. How did that do?
Adenovirus
is nothing.
It's bad. No, it's still. If you eliminate the others to get the scale right, it's still bopping around.
The scale, 2.5% now for ad, and what was rhino?
It was 4% before the pandemic, 15. And it's like 2.5 now. And rhino is
rhino's
doing better.
Rhino is much better. I don't know if it's just the structure of the particle, though. It would be, that could play a role, right? It could be more stable without an envelope. Yeah.
What a bunch of nerds, you guys.
Cool.
But this is so cool.
Hey, it's good. Cool. It's good stuff. You can go back. You can look at other panels too, right? You could look at GI panels. There's a similar graph for that where they have, a lot of bacteria and rotaviruses, astroviruses, adenovirus, noro, sapovirus, parasites too. Wow. Anyway, that's cool. Let's see how noro is doing. Yeah, noro is like 10% of 10% detection rate in GI stuff this year. So that hasn't gone away. So that's oral fecal, and a mask ain't going to help, I guess. When you go to eat, to eat.
So it plummeted right during the lockdowns and is just finally coming back up as people are probably eating out more and traveling more.
Maybe. You have to eat, right?
Yeah.
Anyway, it's a very fun site. You can spend some time.
Good.
There you go. All right, that'll do it for TWiV 739. The show notes are at microbe.tv/TWiV. Questions and comments go to twiv@microbe.tv. And your donations, which help to keep us going, microbe.tv/contribute. Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove's at alandove.com. Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.